Hello everybody. Today let's talk about microorganisms or microbes. These are organisms that we can't see with our naked eye. We require a lens, in other words a microscope, to see them. Now these microbes, at the very outset, let's say only a small proportion of microbes are pathogenic, which means only a small proportion cause disease and even death in plants and animals. Most of them don't harm us. Many of them even help us. And by some estimates, there are over 1 trillion species of microbes on Earth. If you've gone to your grocery store or you just did a search containing the word probiotic, you've probably seen products like this that say that they're great for our gut health because they help the bacteria in our guts. And here's probiotic supplements. I see this culturelle a lot when I go to Costco. There are articles about foods to help promote gut health, dairy foods and fermented foods. I see these different yogurt brands kind of fighting for which is going to be the best for your gut health. So as a society, I think we're more and more conscious that microbes live all over our bodies, inside our bodies, and that they can do great things for us, even if we're not exactly sure what those great things are. Here's a paper in a scientific journal, Life in a World Without Microbes. And it uses a great umbrella term here, microbial services. So we can lump all the good things that microbes do for us under this umbrella term called microbial services. And this paper asks, without microbes and the services they provide, would we die? And it says, no, we wouldn't die, but the quantity and quality of our life would be reduced drastically. So we might not live as long, and while we were living, it would be a pretty terrible life. Here's an example they give of a microbial service. It starts, who would want to live inside a bubble? What they're saying here is that, just as an example of one service, good microbes train our immune systems to protect us against bad microbes. If our immune systems never received this training service, we'd have to live our lives in a bubble because we wouldn't be able to take the onslaught of bad microbes. So that's a pretty bad consequence of not getting this service of immune training, to have to live inside a bubble. There are, this is a great scientific paper. It also talks about, not having microbes, and then you have a poorly developed immune system, you have reduced organ sizes, smaller heart, lungs, liver, you have neurophysiological health problems, you have mental health problems. So microbes are incredibly important for us, for animals. Here's a photo. Here is a photo I took today of my Dendrobium moscatum. It grows very long and it produces a musky fragrance. Now let me ask, do you think that microbes live all over us and in us and help us, but they don't live on our orchids, in our orchids, and help our orchids? And by in our orchids, I mean there have been studies to show that there are microbes living between the cells, between the root cells in orchids. But of course, if these microbes are helping us and living all over us, well, they're living all over our orchids and they're helping our orchids. So here's my moscatum, and then here is, this right here is a little keiki growing from one of the long canes, and these roots are aerial roots. They're touching no substrate. And then here is the medium I'm keeping my moscatum in. It's pine bark, it's Orchiata, and there's a little substrate root. It's poking out, it's surrounded by substrate, and this is some sphagnum moss and some pine bark. I chose to show those pictures of a Dendrobium moscatum because look at this article here, bacteria associated with the roots of epiphytic orchids. This person, her last name is Tsavkilova. Well, she is one of the only people, and this is one of the few papers I've seen devoted to research on microbes on epiphytic orchids. I mean, that is such a niche topic. You can't expect that many people to be researching this, and there's just not that much research on this. But she does prove to you, I mean, she uses a scanning electron microscope, and she has found on the aerial roots of her moscatum 5 different genera. And then she has found on the substrate roots 5 different genera of bacteria. And then on the pine bark substrate, 4 different genera of bacteria. So we know, from her research that there is bacteria growing all over her orchid. And then it's kind of poetic how she describes, what she sees, how these bacteria are growing. I mean, what does it look like? Well, she writes, the excretion of an intercellular matrix, most often of a carbohydrate nature, by the bacteria isolated, confirms its crucial role in the formation of associations of microorganisms with the host plant, particularly in the bacterial attachment to the root surface, which we showed previously with scanning electron microscopy. Bacteria form communities on the roots of tropical epiphytic orchids by means of the matrix. So what it's saying is that these bacteria, what do they look like when they're attached to the matrix? Well, they create this film, and they live in the film, and they're stuck in the film, and the film is stuck on the orchids. So, I mean, here's a good picture of biofilm. And here are some bacteria that have created a biofilm. So I suppose maybe it looks something like this. So, I mean, how interesting that these microorganisms are on the orchids and she sees them. Now, the researcher has proved that there is a microbial community on orchids. But is this microbial community helping the orchids? Well, she says yes, at the very beginning of her paper. At the present time, microbial interactions with plants are receiving more and more attention. Associative microorganisms have a great and often favorable impact upon plant development due to fixation of atmospheric nitrogen, phytohormone synthesis, improvement of water regime and mineral nutrition, and production of fungicidal and bactericidal substances, reducing the number of phytopathogens. Associative microorganisms are considered to attach to the root surface with an intercellular matrix. This researcher's paper was more about describing the microbial community on orchids and not about the services they provide. So let me go more in depth on this with another paper devoted to exactly how microbes benefit plants. So this scientific paper here is called Plant Growth Promoting Bacteria, Mechanisms and Applications. They will abbreviate plant growth promoting bacteria with PGPB. Now, I really like this paper because it tells exactly how microbes, specifically bacteria, help plants. And they actually detail 16 ways that microbes help plants. So they detail 16 microbial services. They organize it into 3 sections. There are direct ways that microbes help plants, and I will just go over these really quickly. I'll just read it. Facilitating resource acquisition. So under that, there is nitrogen fixation, there is phosphate solubilization, there is sequestering iron. Other direct ways that microbes help plants, they modulate phytohormone levels. Some, I guess, phytohormones include cytokines and gibberellins, and something about indoloacetic acid, just something with ethylene, just various direct ways that microbes help plants. But then in this section, I think is really interesting. There are the indirect ways that microbes help plants. It's saying, the good bacteria, they fight the bad bacteria and the bad fungi, and they directly fight those, and then indirectly, because they're destroying the bad, bacteria and bad fungi, it's helping the plants. Read this, antibiotics and lytic enzymes. The synthesis of a range of different antibiotics is a bacteria trait that is most often associated with the ability of the bacterium to prevent the proliferation of plant pathogens, generally fungi. Many of these antibiotics, together with their specificity and mode of action, have been studied in detail, and some of these biocontrol strains have been commercialized. And yeah, look at this University of Massachusetts Amherst, and you have all of these bacteria, Streptomyces lyticus, Bacillus subtilis, that fight fungal diseases, mildew, Pythium, Fusarium, Botrytis. So there are a lot of bacteria that fight fungi. And so yes, the bacteria fight the fungi by synthesizing a range of antibiotics. Some produce enzymes, including, chitinases, cellulases, that lyse a portion of the cell walls of many pathogenic fungi. And they synthesize one or more of these enzymes. Bacteria that synthesize one or more of these enzymes have been found to have biocontrol activity against a range of pathogenic fungi, including Botrytis and Sclerotium and Fusarium and Rhizoctonia, hard to pronounce. And then besides the antibiotics they produce and the enzymes that lyse the cell wall of fungi, they also employ siderophores that they produce. And the siderophores, what it's saying is they have great affinity for iron, more so than fungal pathogens, so that the fungal pathogens are unable to proliferate in the rhizosphere of the roots of the host plant because of a lack of iron. In this model, the biocontrol bacteria effectively outcompete fungal pathogens for iron. So they're taking the iron, so the fungi don't have enough iron. And then in a more generally just general competition, although it is difficult to demonstrate directly, some indirect evidence indicates that the competition between pathogens and non-pathogens, the good bacteria, can limit disease incidence and severity. For example, abundant non-pathogenic soil microbes rapidly colonize plant surfaces and use most of the available nutrients, making it difficult for pathogens to grow. So they can just, colonize the plant surface and then fungi can't be there. And then again, ethylene has some indirect benefit, and there's some induced systemic resistance. And this section is all about how the good bacteria can modulate environmental stress. Again, something to do with ethylene and some IAA and something with cytokinin and something with trehalose and even antifreeze. I think this has to do with the bacteria produce antifreeze so they don't freeze, and because they're all over the plant, it helps the plant not freeze. There are so many services that a microbial community can offer its host plant. This paper has introduced 16 services. As orchid growers, we probably want the microbial community on our orchids that provides the best services and doesn't do anything harmful. However, how microbes interact with one another and then how they interact with macroscopic organisms like us is very complex. We don't know what the best community structure is. Here's my Dendrobium wassellii. Does anybody know what the best microbial community for this Dendrobium is? Do we need X percentage of this bacteria and Y percentage of that bacteria? And the amounts are this and that? Do we even know that there is such a best community? There is such a thing as the best community structure. It probably changes. Probably this is good for winter and that is good for summer. Basically, no one's going to do the research on this, and there's probably, not a single answer. We don't know what the best microbial community is, but what we can answer is that we know what features a really great microbial community will have. Look at the title of this scientific paper. Bacterial Diversity Stabilizes Community Productivity. We know that in all communities of living things, a healthy community is a biologically diverse community. You want zebra, wildebeests, impalas, gazelles, lions, leopards, hyenas, hunting dogs, water buffalo, elands, storks, meerkats, dung beetles, elephants, hippos, rhinos. You want all of the animals in the Lion King. You don't just want to see wildebeests. So what gives you a diverse and abundant microbial community? Well, you want a lot of materials containing carbon. Why? Because most microbes are heterotrophs, meaning they eat carbon. 95% or more of all types of living organisms are heterotrophic, including all animals and fungi and most bacteria and protists. Take a look at this research paper, a preliminary research on the properties of lightweight expanded clay aggregate. So it's a paper on Leica. Now if you go down in the paper, you can see that they give the chemical composition of two different kinds of clay that are used to make Leica. And you can see the clay contains 8 chemical compounds consisting of 9 elements. So the elements are silicone, aluminum, iron, calcium, magnesium, titanium, sodium, potassium, and oxygen. Do you notice what's not there? There's no carbon. Yes, Leica is an inorganic medium, meaning it does not contain carbon, which is what microbes eat. What else might you want for a diverse and abundant microbial community? Well, you don't just want to feed the microbes carbon in one form. You want to feed them carbon as part of a variety of compounds. So, for example, you don't just want to give them glucose and have them derive all their carbon just from glucose. Here is a research paper that, well, calls for more research, like many research papers do. But you can read the highlighted part here. It assumes different microbes. This paper assumes that different microbes are better at breaking down different compounds to get to the carbon. So you want to provide many different sources of carbon so that each microbe can have its niche. So maybe this is really good at breaking down glucose, and that's really great at breaking down lignin. Here's another paper, and it shows a diversity of organic compounds, compounds containing carbon. There's glucose, xylose, alanine, cellulose, starch, pectin, lignin, chitin, tannic acid. Now, what this paper says is that if you give it more diverse inputs, the bacteria community produces more diverse enzymes. So there's a great enzyme diversity with a diversity of organic inputs. Taken together, these results suggest that the role of the microbial community in the decomposition of organic matter is highly dependent on the composition and complexity of inputs to soil. Let's take a look at this book. It's called The Herbal Guide, Dietary Supplement Resources for the Clinician. On page 170, it says, root-based and bark-based botanics also have a broad chemical diversity, with turmeric root and cinnamon bark possessing greater than 1,600 and 1,400 compounds respectively. Now, I already showed that clay that's fired to produce leca contains 8 chemical compounds. 8 chemical compounds. So compare that with what you see here. Turmeric root, just the root, 1,600 chemical compounds. Cinnamon bark, just the bark, 1,400 chemical compounds. So when we talk about a botanical substrate such as pine bark or sphagnum moss, here are some other botanicals. Nettle leaves, over 500 compounds. Rice bran, over 2,600 compounds. When we talk about botanical substrates like pine bark or sphagnum moss, we're talking about hundreds or even thousands of chemical compounds, many containing the element carbon. I don't have a scanning electron microscope or the skills to study a Dendrobium moscatum grown in leca. But does anyone think that an orchid growing with 8 chemical compounds, none of which contain carbon, is going to have the same microbial community structure as one growing with hundreds or thousands of compounds, many containing carbon? This paper, put out by the U.S. Department of Agriculture, titled Effects of Soil Substrate Quality, Microbial Diversity, and Community Composition on the Plant Community During Primary Succession, it says that grasses were not stimulated by microbial diversity, while herbs responded positively to microbial diversity. I'll end by saying, most likely, not every orchid species is so sensitive to the structure of their microbial community. Whether there are a lot of microbes or not, whether the community is diverse or not, what is the dominant species, what is not the dominant species. The problem is that you don't know which orchids are sensitive to the structure of their microbial community. At the very, very end, I'm going to say that I grow my fowls in Leica. I don't live with my fowls. I keep them at my parents' house so that they can enjoy the long-lasting blooms. And because I'm very sporadic with watering them, it makes sense to grow them in Leica versus bark. However, I don't think that they have a great microbial community. But maybe they're not that sensitive to their microbial community. What I think is, if you think that you have a really messed up microbial community structure on your orchid, and so that community is not providing adequate microbial services to your orchid, and your orchid is suffering because of a lack of adequate microbial services, here's something that I thought of that might help. Try incorporating a seaweed-based fertilizer if you don't use one. That's one on the right. You will be adding hundreds of chemical compounds, many containing carbon, and maybe you still get to keep your medium that doesn't break down. Maybe you can have the best of both worlds. Maybe you'll also experience some algae problems. I don't know, but that's it for this video. Take care and happy growing.